Hi, my name is Corinne Smith, and today I will be highlighting my work with the Harris Lab at UC, developing nerve guidance conduits for peripheral nerve injury treatment using piezoelectric scaffolds. So the peripheral nerve system encompasses all of the nerves in your body, excluding your brain and spinal cord. And each year, about 67,000 Americans will suffer from injuries to these peripheral nerves. The potential with the peripheral nerve system is that, unlike in the central nervous system, peripheral nerves have some regenerative capacity, and this is due to the presence of a type of glial cell within the peripheral nerve system called a Schwann cell, which, upon nerve injury, will transdifferentiate into a regenerative phenotype and guide axons across a nerve lesion. So despite this inherent regenerative capacity, for more serious nerve injuries, clinical solutions such as nerve grafts may be required to achieve functional recovery. One new alternative treatment that has been gaining focus is the use of a nerve guidance conduit, which can be used for guiding Schwann cells and consequently axons across a nerve injury. So this is where the work of the Harris Lab comes in, where we are developing piezoelectric nerve guidance conduits to promote regenerative Schwann cell and consequently regenerative nerve cell behavior. And we are doing this using specifically polyvinylidene fluoride trifluoroethylene, otherwise known as PVDF, which is a piezoelectric copolymer. The piezoelectricity of the material is significant in that piezoelectric materials, upon undergoing mechanical deformations, will generate an electric charge, and this electric charge can be used for stimulating cell proliferation and differentiation. So the project I'll be focusing on specifically is the development of a piezoelectric PVDF TRFE fiber scaffold for guiding cell adhesion, proliferation, and alignment. So this scaffold will ultimately be used as the building block for one of these nerve guidance conduits. So the overall goal of this project was essentially to characterize the mechanical and electrical properties of the scaffold, as well as investigate the interactions of the polymer and both Schwann cells and fibroblasts in order to assess biocompatibility. So my involvement with the project was looking at scaffold thickness for mechanical properties, so measuring the thickness of scaffolds for different electrospinning times, since electrospinning was the fabrication method used for the scaffold. And I was also looking at the cell polymer interaction assays, so specifically looking at adhesion and alignment of cells on the scaffolds. So the results of these experiments were that first, for scaffold thickness, increased scaffold thickness increased with longer electrospinning times. And this is significant because increased scaffold thickness can provide additional structural stability within the scaffold. For cell adhesions, cells adhered to all scaffolds, but adhesion was increased with the use of the extracellular matrix protein laminin. And this is significant because it shows that the addition of hydrophilic materials, such as laminin, can be used to promote cell adhesion on an inherently hydrophobic surface like this polymer. For cell alignment, aligned fibers promoted the alignment of both Schwann cells and fibroblasts, and this is consistent with previous works in this field. So overall, the results of this project illustrated that the PVDF TRFE scaffold possesses acceptable mechanical and electrical properties, and it was also illustrated to be biocompatible. So now that the mechanical, electrical, and biocompatible properties of the scaffold have been characterized, the next step will be to develop an actuator for measuring the piezoelectric output of the scaffolds in order to essentially harness the piezoelectric characteristics of this scaffold to influence Schwann cell behavior. And once the relationship between piezoelectricity of the scaffold and Schwann cell behavior has been characterized, the final step will be to use this piezoelectric scaffold to develop actual nerve guidance conduits. So once these nerve guidance conduits have been developed, this work holds potential for developing safer and more effective treatments for peripheral nerve injuries. So here are my acknowledgements. Thank you very much.